So as a graduate student who works on herpes, I'm glad my PI doesn't want to test things on us. First of all, I'd like to thank the speakers for the chance to speak here today. And the question that I became really interested in as a graduate student is how are viral transcripts translated by the host translation machinery despite often looking wildly different than their host counterparts? And we've learned a lot about this from the study of RNA viruses. And some classical examples include the discovery of internal ribosome entry sites that bypass the requirement for a 5' mRNA cap, frameshift elements, which allow for the expression of two proteins from a single mRNA, the production of subgenomic RNAs, or the translation of a large polyprotein, which is subsequently cleaved into the functional proteins. However, fewer of these mechanisms have been discovered in DNA viruses, partially because they tend to replicate in the nucleus and tend to generate mRNA which more closely resemble host transcripts. So in the Kronstad lab, we studied KSHV, which is a large double-stranded herpes virus that Don Gano spoke about earlier today. An analysis of the genome revealed that there was many fewer poly-A sites than the total number of open reading frames. So the number of genetic loci, some of which are indicated here, that give rise to mRNA with the coding potential for more than one protein, or what we call a polycistronic mRNA. So although this subset of viral mRNA looks similar to host transcripts in that they have a 5' cap and a poly-A tail, the presence of multiple open reading frames highlights the question, how does the translation pre-initiation complex access those star codons that are distal from the cap? And in a number of these transcripts, they either use a nested promoter, which give rise to secondary transcript, or use alternative splicing to reposition these downstream genes near to the cap. However, there's a number of these transcripts where neither of these mechanisms are at play, and the strategy remains unknown. So one such transcript encompasses ORF35 to 37, which was first characterized by Hackenau back in 2006. And while we identified, and this encompasses 35 to 37, and while we identified a nested promoter within ORF36 that gives rise to a 37 specific mRNA, importantly, there was no transcript in infection where ORF36 was present as the 5' gene. So we asked the question, how does the translation pre-initiation complex access the ORF36 star codon? And we were interested in this just from a basic gene expression standpoint. We were also interested because ORF36 encodes for the viral protein kinase. And this is a protein which is known to phosphorylate and activate antiviral prodrugs such as goncyclovir. And this creates a monophosphate intermediate, and then cellular kinase finish the job creating an active drug. And importantly, treatment of patients with goncyclovir delays the onset of Kaposi sarcoma. So one of the first questions we asked is whether this polycistronic mRNA can support translation of both 35 and 36. Is this a functional bicistronic transcript? So to look at this, we engineered a construct with an N-terminal tag on 35, a C-terminal tag on 36, while maintaining the native 5' UTR. And we in vitro transcribed this mRNA and expressed in cells. And here you can see 35 alone, 36 alone, and importantly, expression of both proteins from a single mRNA, confirming this is bicistronic, and intriguingly, expression of ORF36 was robust despite being present as the downstream gene, which is really not what we expected. So we asked whether expression of 36 was dependent on scanning from the 5' mRNA cap. And to look at this, we inserted a very strong hairpin within a native 5' UTR, which is known to impede linear scanning. And this background, we saw that expression of both 35 and 36 was strongly compromised. And consistent with this data, when we directly tested the sequences upstream of 36, there was no detectable internal ribosome entry site activity. So since scanning from the cap is important for 36 expression, we expected that the ORF35 star codon should be strongly inhibitory to 36 expression. So we weakened the Kozak consensus sequence surrounding this AUG, and predictably expression of 35 goes down. We were surprised to see a negligible effect on 36. So this would argue against a leaky scanning or frameshift mechanism and further indicates there may be a mechanism in place which disfavors initiation at the ORF35 star codon. So we searched with features within the 5' UTR, which is a traditional hotbed of regulatory elements, and we identified two short upstream open reading frames. So just a brief primer into uORF biology. Their functions as global regulators of gene expression. They function by dampening translation of the primate ORF. And they do this by forcing the ribosomes to either leaky scan past the uORF star codon or by reinitiating after uORF termination. And this level of repression can range anywhere from 30 to 80%. And there's two critical parameters which influence the efficiency. First, the length of the uORF, and second, the intercistronic distance between the uORF stop codon and the beginning of the primary ORF. So in this mRNA, uORF1 is completely within the 5' UTR. It's 9 codons in length and has a start codon which resides in a weak context. uORF2 is a little bit longer, importantly has an AUG in a strong context, and the positioning of uORF2 indicates that translation of uORF2 and 35 are mutually exclusive events. So we first asked, what was the contribution of uORF1 towards translation of 35 and 36? So we simply disrupted the uORF1 start codon. In this background, we saw a modest increase in 35 expression, indicating that uORF1 is functioning as a canonical cellular uORF by repressing translation of that 5' gene. And importantly, certain uORFs are known to confer susceptibility to certain RNA decay pathways. So we also did a corresponding northern blot, looking at both the transcript, which has similar transcript accumulation, a GFP co-transfection control, and an 18S loading control. So this confirms that this is at the level of the protein and not an effective transcriptional rates or RNA decay pathways. All right, so we next looked at uORF2 and did the same mutation, disrupted the uORF2 start codon. And importantly, we designed these mutations to make sure uORF1 and uORF2 were looked at independently. And in this background, we saw that 35 expression goes up, while 36 expression was compromised. So this indicates that uORF2 is repressing 35 and allowing for the expression of the ORF36 gene. And in data that I don't have time to share with you, we both looked at the uORF length and the intercistronic distance and showed those are critical for reinitiation at the ORF36 start codon. Finally, we wondered what was the role of this uORF in the context of an entire KSHV infection. So look at this, we collaborated with Kevin Brula, a graduate student in Jay Zhang's lab at USC, to make a single nucleotide change in the uORF in the KSHV back system. So we disrupted the uORF and made the corresponding marker rescue. We generated cell lines which each of these 3 backs, which each lytically reactivated as indicated by markers of the ORF57 protein. And while ORF36 expression was readily detectable in the wild type in the marker rescue, there was a strong defect in 36 expression in the uORF2 mutant, confirming that this uORF is critical for the ORF36 encoded viral protein kinase during KSHV infection. We then wanted to look at the entire viral life cycle, so we performed an infectious titer assay, where you take the soup from lytically reactivated cells and infect a new cell line. And since the back constitutively expresses GFP, you can readily detect the drop in infectious titer between the marker rescue and the uORF2. And in flow analysis, we quantified this to be a 37-fold drop in infectious titer. Finally, we wondered if these uORFs were specific to KSHV or whether they were conserved within the gamma herpes urinase subfamily. And indeed, we identified a uORF present upstream of the ORF35 homologs in Epstein-Barr virus, the primate herpes virus Semeri, and the spider monkey Adeline herpes virus 3. We also identified a uORF overlapping the ORF35 homologs in those 3 viruses, as well as an additional rhesus radenovirus. And although we don't yet know if these uORFs are functioning similarly in these viruses, it was interesting to note that there was no obvious amino acid conservation, indicating that these uORFs are likely functioning simply by engaging the translation initiation machinery. So collectively, our data support a model where KSHV has stolen a pathway from cellular mRNA, normally used to dampen translation of the primary ORF, and repurposed it to allow for the ribosomes to shuttle past the start codon of the 5' gene to allow for the translation of the downstream gene. And while this may simply be an elaborate mechanism to conserve coding space, we're currently investigating whether these uORFs serve as some sort of translational switchboard, allowing for the expression of these proteins either at different points during the viral life cycle or under certain cellular conditions. And excitingly, we've identified a second KSHV bistrong mRNA that harbors a uORF in its 5' UTR. And we're excited to see how widely spread this mechanism occurs in nature. And with that, I'd like to thank my fantastic professor, Britt Glonsinger, who doesn't use us for experiments, and all the members of the Glonsinger Lab, our kind collaborators, Dr. Jay Zhang and Kevin Brula, these professors for reagent gifts, as well as Burroughs Wellcome and NSERC for funding. And I'd love to answer any questions that you may have. Yes, Dr. Vance.
Very nice story. So if I understood correctly, so the purpose of, well, ORF36 has some role in the viral life cycle that's essential, obviously.
Yes.
But it also makes the virus susceptible to ganciclovir. Is that right?
Correct, yes. Mm-hmm.
So have you, this is kind of a crazy idea, but have you investigated whether, I mean, in the presence of ganciclovir, wouldn't it be advantageous to inactivate that upstream ORF? So have you looked at whether perhaps that might be functioning in some way as a sensor of ganciclovir? Is that a crazy
The
idea?
sensor of ganciclovir.
Well,
We
'cause
haven't
would you
done-
want to turn off ORF36 in the presence of ganciclovir? So then you would want to inactivate the upstream ORF, if you were the virus, I'm thinking from the virus's point of view.
Perhaps, and I don't know of any of those that happening in KSHV, but there are studies with the thymidine kinase protein, which performs the same function in alpha herpes viruses, and there are mutant herpes viruses that have deactivated thymidine kinase, which make it no longer susceptible to the related acyclovir drugs. So it definitely would be something that the virus could use, yeah. But I mean, obviously you see here that there's a strong defect in the viral replication, so it would be that trade-off cost-benefit analysis of the virus, yeah. And actually, since ganciclovir is not used as a therapeutic treatment, usually people who have Kaposi's sarcoma are treated with HAART, so it wouldn't be that selective pressure wouldn't be there, yeah.
Do you know if there are any long-range interactions in this Polycistronic messenger RNA?
None that we know of, and we know the 3' UTR is very short in this mRNA, so it's not like there's this long, several hundred nucleotide 3' UTR which is present, which might contribute to these long-range interactions, but none that we know of so far.